Orchestrated Objective Reduction, the Penrose-Hameroff model of quantum consciousness, proposes that quantum processes in neuronal microtubules underlie subjective awareness. Roger Penrose, the Nobel Prize-winning mathematical physicist, and Stuart Hameroff, the anesthesiologist, have developed this framework linking consciousness to space-time geometry itself, making awareness not merely biological but fundamentally cosmic in nature. Quantum biology in ambient conditions. Anesthesia mechanisms at the molecular level. Objective collapse models in quantum measurement. Biophotonics and ultra-weak photon emission from biological matter. Neural synchronization and the gap between correlation and entanglement. These are not speculative fantasies. They are active research programs with laboratory data from researchers like Jack Tuszynski at the University of Alberta and Gregory Scholes at Princeton. The question is not whether quantum effects exist in biological systems. They demonstrably do. The question is whether those effects have anything to do with consciousness. And between 2022 and 2024, new measurements arrived that changed the shape of this debate. The evidence file on quantum consciousness contains multiple categories: photophysical measurements in purified microtubule preparations, behavioral anesthesia experiments in rodents, contested MRI signals claiming non-classical brain function, and theoretical models connecting consciousness to space-time geometry through gravitational self-energy. Each category carries its own burden of proof, its own methodological constraints, its own published disputes. This stands in contrast to competing frameworks like integrated information theory, developed by Giulio Tononi, which treats consciousness as emerging from classical information integration, or global workspace theory, which explains awareness through distributed neural processing without requiring quantum mechanics. What follows is a forensic examination of that evidence, not interpretation, not advocacy. The data itself, the measurements, the instruments, the published disputes, and the implications, some verified, some contested, some sitting at the boundary of what current methods can resolve. By the end of this video, you will understand what Orch OR actually claims at the minimum testable level, what the 2022 to 2024 experiments measured, why anesthesia remains an incomplete mechanistic puzzle, how decoherence constrains feasibility, and where the genuine scientific disagreements lie. You will understand the difference between demonstrating quantum effects in biological systems and demonstrating that those effects matter for consciousness. You will understand the scale problems, the gap between nanometer molecular structures and millimeter MRI voxels, between femtosecond coherence times and millisecond cognitive processing, between purified laboratory preparations and living neuronal tissue. If this channel is the kind of content you return to, consider subscribing. There is no shortcut for depth. Let us begin with what orchestrated objective reduction actually asserts, stripped of mysticism, reduced to testable propositions. Orch OR, in its minimal form, makes three linked claims. First, microtubules in neurons support functionally relevant quantum states, coherence, entanglement, or collective modes under physiological conditions. Warm, wet, noisy. The environment that ordinarily destroys quantum coherence in laboratory systems. Orch OR claims microtubules maintain it. Second, anesthetics abolish consciousness in part by disrupting those microtubule quantum states. This would explain the selective loss of conscious awareness under general anesthesia, while many non-conscious functions, respiration, circulation, reflexes, continue. The selectivity is the clue. something specific to consciousness is being targeted. Third, a distinct collapse-like step, objective reduction, terminates each orchestrated quantum episode on a timescale set by a gravitational self-energy threshold. The equation commonly summarizing this is tau approximately equals h bar over E sub g. Tau is the collapse timescale. H bar is the reduced Planck constant, approximately 1.055 times 10 to the negative 34 joule seconds. E sub g is the gravitational self-energy of the superposed mass distribution. This third claim makes Orch OR distinctive. It ties consciousness to space-time geometry through the quantum measurement problem, not through mystical connection. Through a proposed physical mechanism that Roger Penrose, the mathematical physicist, developed from considerations about computability and the nature of understanding. The gravitational self-energy component deserves careful attention. When matter exists in quantum superposition, occupying two or more configurations simultaneously, the mass distribution creates a corresponding superposition of spacetime curvature. Under general relativity, spacetime curvature is not fixed. It responds to mass. Superposed mass means superposed spacetime. Roger Penrose proposed that this superposition is inherently unstable, that it triggers objective collapse on a timescale inversely proportional to the gravitational self-energy difference between configurations. This is not proven. It is a hypothesis with mathematical structure and, in principle, testable implications. Roger Penrose is not a fringe figure. He shared the 2020 Nobel Prize in Physics for demonstrating that black hole formation is a robust prediction of general relativity. His mathematical work on spacetime singularity theorems, Penrose tilings, and twistor theory established him as one of the most significant mathematical physicists of the 20th century. His contributions to cosmology and the theory of spacetime continue to influence research decades after their initial publication. When Roger Penrose proposed that consciousness relates to quantum gravity through objective collapse, the scientific community could not dismiss him as a crank. They could, and did, subject the proposal to intense scrutiny. The origins of Penrose's interest in consciousness trace back to his work on the foundations of mathematics and computation. In his 1989 book, The Emperor's New Mind, and its 1994 sequel, Shadows of the Mind, Roger Penrose argued that human mathematical understanding cannot be reduced to algorithmic computation. He drew on Gödel's incompleteness theorems to suggest that consciousness involves non-computable elements, processes that cannot in principle be simulated by a Turing machine. This led him to quantum mechanics, where the process of wave function collapse introduces genuine randomness and potentially non-computability. The question then became, where in the brain could quantum processes relevant to consciousness occur? Hameroff, the anesthesiologist who collaborated with Penrose, brought the microtubule component. Hameroff's clinical experience with general anesthesia, drugs that reversibly abolish consciousness while preserving many other functions, led him to focus on what consciousness specifically requires. Ion channels and synaptic transmission underlie neural signaling in general. Anesthetics do not simply stop signaling, they selectively remove awareness. Hameroff proposed that microtubules, the structural polymers running through every neuron, were the site where consciousness-specific processing occurred. Hameroff's background is important for understanding his contribution. As a practicing anesthesiologist, he administered consciousness-abolishing drugs to patients daily. The clinical phenomenon was unmistakable. One moment awareness was present, the next it was gone, and then it returned. This reversible abolition of consciousness, with relative preservation of many other neural functions, suggested something specific was being targeted. Standard explanations focused on ion channels and synaptic receptors. Stewart-Hameroff proposed that microtubules might be an additional or alternative target, and that the quantum properties of these structures might be essential to consciousness. Microtubules are not mysterious structures. They are well-characterized components of the cytoskeleton. Microtubules consist of tubulin dimers, proteins composed of alpha-tubulin and beta-tubulin subunits, arranged into cylindrical polymers with 13 protofilaments in most cells, though variants with 14 protofilaments also exist and have been examined in the recent research. The tubulin dimer dimensions measure approximately 4 nanometers by 6.5 nanometers by 8 nanometers, corresponding to a sphere of diameter roughly 7.4 nanometers, about 2.91 times 10 to the negative 7 inches. These are not exotic molecules. They are among the most abundant proteins in cells. Every cell type in the human body contains microtubules. Microtubules provide structural support, serve as tracks for intracellular transport, and participate in cell division. The question is whether they do something unusual, something quantum, in neurons. The dominant view in neuroscience does not require quantum coherence. Most models of consciousness rely on network dynamics and classical electrophysiology, the firing of action potentials, the summation of synaptic inputs, the emergence of integrated information or global workspace access through distributed neural activity. These classical models are not failing. They explain many phenomena. They guide clinical neuroscience, brain imaging interpretation, and understanding of neurological disorders. They do not fully explain one specific phenomenon, what anesthetics actually target to abolish consciousness. The Meyer-Overton correlation, discovered over a century ago, remains relevant to this discussion. In the late 19th and early 20th centuries, researchers noticed that anesthetic potency correlates with solubility in a hydrophobic medium, specifically olive oil. The more lipid-soluble a compound, the more potent it tends to be as an anesthetic. This correlation spans many orders of magnitude and many different chemical structures. The Meyer-Overton correlation suggests that anesthetics work through relatively non-specific interactions with lipid membranes or hydrophobic protein pockets rather than through precise lock-and-key receptor binding. It does not identify the specific molecular target. Many molecular sites are hydrophobic. Lipid membranes, ion channel interiors, protein-binding pockets, microtubule interior spaces. The lack of a single dominant molecular target for anesthesia is relevant to the Orch OR discussion. If there were one clear receptor, like morphine binding to opioid receptors, the mechanism would be settled. Anesthesia appears to involve multiple targets, and the specific combination that produces unconsciousness remains debated. This is the part that stays with me, not because it is exciting, because it is heavy. Anesthesia works. Every day in operating rooms across the world, patients receive volatile anesthetics like isoflurane or intravenous agents like propofol, and consciousness disappears. Reversibly, reliably, the engineering works. And yet the precise molecular mechanism by which consciousness is abolished remains described in the scientific literature as incompletely resolved. Multiple targets have been identified: ion channels, receptors, synaptic proteins, mitochondria, and yes, cytoskeletal components. Which combination produces unconsciousness, and through what causal chain, remains an open question. This is an engineering triumph built on an explanatory gap. We know how to abolish consciousness. We do not fully understand why it works. Now let us examine what the 2022 to 2024 experiments actually measured. In March 2023, a paper titled Electronic Energy Migration in Microtubules was published. The author list includes Jack Tuzinski, a biophysicist at the University of Alberta, who has contributed extensively to computational modeling of microtubule properties, along with Arat Kalra and Gregory Scholes at Princeton. Gregory Scholes is a prominent chemist whose laboratory specializes in quantum effects in energy transfer. What did they measure? The researchers used tryptophan autofluorescence to probe energy transfer within microtubules. Tryptophan is an amino acid that absorbs ultraviolet light and emits fluorescence. It serves as a natural chromophore, a light-absorbing unit embedded in the protein structure. By measuring how fluorescence lifetime changes when an external quencher is introduced, researchers can infer how electronic excitation energy migrates through the system. The instrument chain matters for understanding what was actually observed. UV-Vis spectrometry using a Carry 6000I with integrating sphere for absorbance measurements. Steady-state fluorescence using a PTI Quantumaster spectrofluorometer. Time-correlated single-photon counting TCSPC using a DeltaFlex system with 305 nanometer LED excitation and detection at 335 nanometers. Transmission electron microscopy using a thermoscientific Talos L120CTEM at 200 kilo electron volts for structural characterization. These are not exotic instruments. They are standard spectroscopy tools. What was unusual was the result. The reported diffusion length for electronic excitation in microtubules measured 6.64 plus or minus 0.1 nanometers. This is comparable to the size of a single tubulin dimer. The energy does not stay localized. It migrates, hopping from chromophore to chromophore across a distance roughly equal to 1 protein unit. To put this scale in perspective, if the tubulin dimer diameter of 7.4 nanometers was scaled to 1 meter, approximately 3.28 feet, the diffusion length of 6.64 nanometers would scale to approximately 0.90 meters, or about 2.95 feet. The excitation travels almost an entire dimer length before decaying or being quenched. This is dimer-scale transport, not trivial, but also not extending across the entire microtubule polymer. Why does this matter? The baseline prediction from first-resonance energy transfer, the standard model for dipole-dipole mediated non-radiative energy transfer, underpredicted the observed diffusion length. First theory describes how electronic excitation energy transfers between chromophores through the electromagnetic coupling of their transition dipole moments. The rate depends on the inverse sixth power of the separation distance and on the relative orientation of the dipoles. The first coupling equation in its ideal dipole form can be written as V Coulomb is proportional to mu I times mu j times kappa over 4 pi epsilon naught n squared r cubed. Here, mu I and mu j are the transition dipole moments of the interacting chromophores. Kappa is an orientation factor. Epsilon naught is the permittivity of free space. N is the refractive index of the medium, and R is the separation between chromophores. The authors report that conventional first theory estimates alone, as implemented in their baseline simulations, underpredict what was measured. This implies additional coupling mechanisms beyond the point-dipole approximation may be relevant. Perhaps orbital overlap contributions, exchange coupling at short distances, or collective effects that standard theory does not capture. The Stern-Volmer analysis used to infer transfer rates relates quenching to concentration. Tau naught over tau equals 1 plus k sub SV times quencher concentration. Here, tau naught is the fluorescence lifetime without quencher, tau is the lifetime with quencher, and k sub SV is the Stern-Volmer quenching constant. By measuring how lifetime changes with added quencher, the researchers extracted diffusion parameters. The diffusion length equation is L equals the square root of 4d tau. L is the diffusion length. D is the diffusion coefficient. Tau is the characteristic lifetime or time window. This converts measured lifetime and quenching data into a nanometer scale transport length. Now here is where anesthesia enters the picture. The researchers introduced two anesthetics, etomidate and isoflurane, at a concentration of 50 micromolar. Both reduced the measured exciton diffusion length. Etomidate reduced it from 6.6 plus or minus 0.1 nanometers to 5.6 plus or minus 0.1 nanometers. Isoflurane reduced it to 5.8 plus or minus 0.2 nanometers. Think about that measurement. Let it settle. A general anesthetic, one that abolishes consciousness in living organisms, measurably reduces energy transfer in a purified microtubule preparation. This does not prove that consciousness depends on microtubule energy transfer. It does not prove that anesthetics act through this mechanism in living brain tissue. It establishes a biophysical connection between anesthetic molecules and microtubule photophysics that exists and can be measured. The paper itself is careful about interpretation. It notes that cells are unlikely to utilize the light-harvesting properties of tubulin in a straightforward sense. The UV excitation used in the laboratory is not present at physiological light levels inside neurons. The electronic coupling that enables energy migration could have other functional manifestations, perhaps in how microtubules process or transmit information through conformational or electronic states. What the paper establishes is a mechanistic substrate. Microtubules can exhibit unexpectedly long-range energy migration. Anesthetics measurably reduce it. Whether this connects to consciousness remains to be determined. In May 2024, a second paper appeared. Ultraviolet superradiance from meganetworks of tryptophan in biological architectures. This paper, published in open access format and associated with researchers at Howard University in Washington, D.C. and collaborators, extends the quantum optical analysis to collective emission effects. Superradiance is a phenomenon first described by Robert Dicke in 1954. When multiple emitters, atoms, molecules, or chromophores are coupled together within a wavelength of the radiation they emit, their decay rates can be collectively enhanced. A group of n-coupled emitters can emit photons at a rate proportional to n squared rather than simply n. This is a coherent enhancement arising from the phase relationships between emitters. The physics is subtle but well established. Individual emitters, acting independently, would produce radiation at a rate proportional to the number of emitters, n. If the emitters are coupled through their shared electromagnetic field, certain collective states become bright. They decay faster than individual emitters would. This is superradiance. The enhancement arises because the amplitudes of emission from different emitters add constructively. The opposite effect, subradiance, also occurs. Some collective states have suppressed decay rates, becoming dark and long-lived. In these states, the emission amplitudes add destructively, reducing the overall decay rate. A system with superradiant and subradiant states can have coexisting timescales spanning many orders of magnitude. This has implications for quantum information. If subradiant states are truly long-lived, they could provide a mechanism for quantum coherence to persist longer than the decoherence time of individual chromophores. The coherence would hide in the dark states, protected from rapid decay. The 2024 paper models arrangements of up to more than 10 to the fifth power, more than 100,000 tryptophan transition dipoles in microtubule architectures. The excitation wavelength used was approximately 280 nanometers, the natural absorption peak of tryptophan. This is ultraviolet light, with energy around 4.4 electron volts per photon. The instruments used for the experiments included Shimadzu RF5301PC for fluorescence spectroscopy and Shimadzu UV3600 for absorption spectra. These are standard research-grade spectrometers capable of precise measurement in the ultraviolet range. What did they find? The authors predict strongly superradiant states from collective interactions among organized tryptophan arrays. They state this leads to enhancement of fluorescence quantum yield that is confirmed by our experiments. The quantum yield, Qy, is the fraction of absorbed photons that result in emitted photons. It is calculated as Qy equals gamma over gamma plus gamma sub nr. Gamma is the radiative decay rate. Gamma sub nr is the non-radiative decay rate. For reference, the radiative lifetime for tryptophan in isolation is approximately 1.9 nanoseconds, and the quantum yield in water is estimated around 0.13, meaning only about 13% of absorbed photons result in fluorescence, with the remainder decaying through non-radiative pathways. The predicted timescales are striking. Bright states, the superradiant collective modes, have lifetimes in the hundreds of femtoseconds, around 10 to the negative 13 seconds. Dark states, the subradiant modes, have lifetimes in the tens of seconds, around 10 to the 1 seconds. That spans approximately 14 orders of magnitude in lifetime within the same theoretical framework, coexisting in the same molecular architecture. Let me emphasize this span. 100 femtoseconds, 10 seconds. The ratio between them reaches 10 to the 14th power, 100 trillion times longer. The brightest states flash and vanish in less time than it takes light to cross a human hair. The darkest states persist for longer than you have been watching this video. The paper emphasizes that this superradiant behavior persists in the presence of disorder and at thermal equilibrium. This is counterintuitive. Standard quantum optical effects require carefully controlled conditions, often ultra-cold temperatures, purified samples, isolated systems. The claim that biological proteins at room temperature can exhibit collective emission patterns challenges that intuition. Let me be clear about what this paper does and does not establish. It establishes, under their modeling framework and experimental measurements, that quantum yield scaling with tryptophan network size is consistent with a superradiance interpretation. Whether alternative classical or semi-classical interpretations could reproduce similar scaling is an open technical question. The interpretation is contested precisely because the measurements are at the boundary of what current models can uniquely discriminate. What the paper does not establish is any direct connection to consciousness or cognition. it establishes photophysics, how light interacts with microtubule architectures, the link to neural function, to subjective experience, to what happens in a living brain during awareness or anesthesia. That link is not measured in this paper. It is hypothesized. In August 2024, a third paper appeared that brings the evidence closer to living organisms. Microtubule stabilizer epothalone B delays anesthetic-induced unconsciousness in rats. This paper represents a different kind of evidence from the spectroscopy studies. It is behavioral. It involves living animals. It measures something closer to consciousness itself, or at least to a standard proxy for consciousness. The experimental design is straightforward but powerful. Take two groups of rats. Give one group a drug that stabilizes microtubules. Give the other group a control treatment. Then expose both groups to anesthetic and measure how long it takes each to lose consciousness. This paper reports an in vivo behavioral experiment. The subjects were male Long-Evans rats, approximately 2 months old, weighing 220 to 266 grams, about 7.8 to 9.4 ounces. The sample size for the main experimental group was N equals 8. The intervention was subcutaneous injection of epothalone B, a microtubule stabilizer, at a dose of 0.75 milligrams per kilogram, approximately 0.34 milligrams per pound. Epothalone B binds to microtubules and enhances their stability, preventing depolymerization. The drug binds to the same site on tubulin as paclitaxel, also known as Taxol, but with better blood-brain barrier penetration. Epothalone B is used clinically in cancer treatment, where disrupting microtubule dynamics interferes with cell division. The anesthetic was isoflurane, a volatile anesthetic commonly used in surgery, delivered at 4% concentration in oxygen at 1.5 liters per minute flow, approximately 0.396 gallons per minute. Isoflurane is one of the most commonly used general anesthetics in both clinical medicine and research. It is inhaled, rapidly absorbed through the lungs, and reaches the brain within seconds of inhalation. The outcome measure was loss of writing reflex, LORR. This is a standard proxy in rodent anesthesia research. When a rat is turned on its back, it normally rights itself immediately. Under anesthesia, this reflex is lost. The time from anesthetic exposure to LORR measures how quickly anesthetic-induced unconsciousness develops. LORR is not a direct measure of consciousness. We cannot know what a rat experiences subjectively. It indexes purposeful, coordinated behavior that requires integration of sensory information and motor planning. When LORR is lost, the animal cannot perform a basic survival-relevant behavior. This is a reasonable proxy for loss of awareness. What did they find? Epothalone B-treated rats took approximately 69 seconds longer to lose writing reflex compared to controls. The effect size was large, Cohen's d approximately 1.9. In statistical terms, Cohen's d greater than 0.8 is considered a large effect. Cohen's d of 1.9 is very large. The distributions of the two groups barely overlap. 69 seconds, a minute and 9 seconds. That is how much longer consciousness persisted when microtubules were pharmacologically stabilized against anesthetic action. To put this in perspective, under the 4% isoflurane protocol used, control rats typically lost writing reflex within a few minutes of exposure. A 69-second delay represents a substantial fraction of the total induction time. The effect is not subtle. The authors note that their result does not specify the mechanism by which microtubule stabilization affects anesthetic sensitivity. Microtubules have many functions: structural support, intracellular transport, signaling scaffolds, cell division. Stabilizing microtubules affects all of these functions. The result is consistent with quantum coherence hypotheses, but it is also consistent with classical cytoskeletal mechanisms. What the result establishes is that microtubules matter. They are not irrelevant to anesthesia. Any complete account of how anesthetics abolish consciousness must include microtubules among the relevant targets. This result tangibly challenges any simplistic account that dismisses microtubules as irrelevant to anesthesia. A pharmacological manipulation targeting microtubules, not membrane receptors, not ion channels, not synaptic proteins directly, changed anesthetic sensitivity under a standard behavioral proxy. The paper does not directly measure microtubule quantum coherence or superradiance in living brain tissue. It supports microtubules as functionally relevant to anesthetic action. It does not establish the mechanism. The 69-second delay could arise from classical cytoskeletal roles: transport, trafficking, structural stability, signaling scaffolds. The quantum hypothesis is not the only interpretation. This brings us to the question of what would constitute a decisive experiment. An ideal experiment would combine behavioral measurements with direct spectroscopic detection of microtubule quantum states. You would measure fluorescence dynamics in living brain tissue while simultaneously measuring anesthetic response. You would show that the quantum states correlate with consciousness, and that disrupting them specifically abolishes awareness. Such an experiment faces formidable technical barriers. Optical access to deep brain tissue at nanometer resolution is not currently possible without substantial tissue damage. The instruments that detect microtubule photophysics, time-correlated single-photon counting, UV spectroscopy work on purified preparations, not intact brain. Bridging this gap requires new techniques that do not yet exist. Before I continue, consider what this means. Take a moment. 3 papers, 3 lines of evidence. Energy migration in microtubules that anesthetics reduce. Collective emission patterns consistent with superradiance modeling. Behavioral anesthesia sensitivity that shifts when microtubules are stabilized. None of these proves consciousness depends on quantum processes in microtubules. Together, they establish that microtubules have measurable quantum optical properties and are relevant to anesthetic action. The question is whether these properties are connected. Do the quantum optical effects measured in vitro exist in living brain? Do they affect neural function? Does stabilizing microtubules change anesthetic sensitivity through quantum mechanisms or through classical ones? The 2022 to 2024 evidence does not answer these questions. It establishes the substrate. Microtubules can do unexpected things with energy and light, and microtubules matter for anesthesia. Whether there is a causal connection between these facts and whether that connection involves quantum physics remains to be determined. This is the state of the evidence, not proof, not refutation. Open inquiry with new tools and new data. This brings us to the contested evidence, the claims that have generated explicit published dispute. In October 2022, Christian Matthias Kerskens and David Lopez Perez at Trinity College Dublin published a paper reporting what they interpreted as evidence for non-classical brain function. They used specialized MRI pulse sequences designed to detect zero quantum coherence, ZQC. To understand this claim, we need to understand what MRI measures and what zero quantum coherence means. Standard MRI measures the precession of hydrogen nuclei, protons, in a strong magnetic field. When placed in a magnetic field, protons align with or against the field and precess around the field direction at a frequency called the Larmor frequency. Radio frequency pulses can tip these spins away from equilibrium, and as they return, they emit signals that form the basis of MRI contrast. This standard signal comes from single quantum coherence, the coherent precession of individual nucleus spins. More complex spin states exist. When multiple spins interact, which they do in molecules, correlated multi-spin states can form. Some of these are called zero quantum coherences. Zero quantum coherence is a class of multi-spin coherence terms in nuclear magnetic resonance experiments. These involve correlated states where the quantum numbers add to zero, hence zero quantum. They do not precess at the Larmor frequency and require special pulse sequences to detect. Ordinary MRI sees single quantum coherences. ZQC requires targeted acquisition. The technical interest in ZQC is that these multi-spin correlations could in principle reflect quantum coherence between spatially separated spin pairs. If such coherence were maintained over macroscopic distances and timescales, it might constitute evidence for non-classical behavior. The experimental setup, 40 subjects aged 18 to 46 years, scanned in a 3 Tesla MRI system, a Philips scanner with 32-channel array coil. The imaging parameters included gradient echo planar imaging with repetition time 45 milliseconds, echo time 5 milliseconds, voxel size 3.5 millimeters, approximately 0.138 inches, flip angle 45 degrees, matrix 64 by 64, sense factor 3, plus saturation pulses and detailed gradient integrals specified in the methods. The repetition time of 45 milliseconds is unusually fast for standard MRI. This rapid sampling allowed the researchers to resolve signal changes on sub-second timescales, including cardiac synchronized variations. What did they claim? The authors report heartbeat-locked signal bursts in the MRI time series with peaks up to approximately 15%. The signal appeared in specific brain regions and showed periodic variation locked to the cardiac cycle. They argue these signals exceed what classical models predict for their pulse sequence. The entanglement witness interpretation works as follows. The researchers computed a classical bound, the maximum signal that should arise if only classical correlations were present. They claim their measured signal exceeds this bound, and under the entanglement witness framework implies the presence of quantum entanglement. They further claim the signal varied with conscious awareness, differing between wakefulness and altered states induced by breath holding or other protocols. This is a strong claim. Entanglement in living brain tissue, measured non-invasively with MRI, correlated with consciousness. If verified, it would be among the most significant findings in consciousness research. The implications would extend far beyond Orch OR. It would demonstrate that macroscopic quantum coherence can survive in brain tissue long enough to be detected by MRI, which operates on millisecond timescales. It was not verified, it was disputed. In March 2023, Warren S. Warren, a chemist at Duke University with extensive expertise in NMR pulse sequences and quantum coherence measurements, published a comment directly criticizing the Kerskens and Lopez-Perez paper. Warren is a leading figure in magnetic resonance methodology. His group has developed advanced pulse sequences and contributed to understanding how multi-spin coherences behave. His critique carries substantial technical weight. Warren's argument: the paper provides no evidence justifying the entanglement claim. The results reflect complexity and MRI limitations rather than demonstrated non-classicality. The critique has several components. First, Warren questions whether the pulse sequence actually isolates ZQC as claimed. MRI signals arise from complex magnetization dynamics involving relaxation, diffusion, flow, and compartmentalization. The claim that a particular signal component represents zero quantum coherence requires careful demonstration that other contributions have been ruled out. Second, Warren challenges the classical bound computation. The bound depends on model assumptions about what classical physics predicts. If those assumptions are incomplete, if they fail to account for some conventional physics mechanism, then exceeding the bound does not imply non-classicality. Warren argues the classical model used was not sufficiently comprehensive. Third, Warren points to practical MRI artifacts. Cardiac pulsation causes brain motion. Blood flow changes with the heartbeat. Cerebrospinal fluid moves. These physiological dynamics can produce time-varying MRI signals that correlate with the cardiac cycle without any quantum effects. The thrust of Warren's critique is that the data show complexity, not non-classicality. The signals are interesting and perhaps poorly understood, but jumping to entanglement is not justified by the evidence presented. The authors replied in the same journal. They defended their classical bound argument, asserting they did not measure standard intermolecular multiple quantum coherence, IMQC, but signals exceeding what they computed as classical thresholds. They addressed some of Warren's specific technical points and reiterated their interpretation. The disagreement remains unresolved in the published record. Neither side conceded. The scientific community has not reached consensus on whether the MRI signals reported constitute evidence for brain entanglement. Vlatko Vedral, a prominent quantum physicist at Oxford, was quoted in a 2022 secondary report on this work. He emphasized that demonstrating such effects in living cells would be significant regardless of links to consciousness. The significance is precisely the point. Extraordinary claims require extraordinary evidence, and the evidence remains disputed. What would it take to prove entanglement in living brain tissue? This is a hard question, and it deserves explicit treatment. In controlled laboratory systems, entanglement is typically demonstrated through Bell inequality violations, correlations between spatially separated measurements that exceed what any local classical theory can explain. Bell tests require precise control over measurement settings and outcomes, typically at the single quantum level. The Bell inequality framework is powerful because it makes minimal assumptions. If you can perform measurements on two spatially separated systems and find correlations that violate Bell's inequality, then, under reasonable locality assumptions, those correlations must arise from entanglement. No classical hidden variable theory can reproduce them. MRI operates at an entirely different scale. A 3.5 mm voxel averages over enormous microstructural diversity, millions of cells, billions of molecules, trillions of nuclear spins. Any claim tying MRI signals to microtubule scale quantum processes must confront this spatial averaging gap. How do molecular scale quantum events produce macroscopic, reliably structured signals in voxels containing so much complexity? The Kerskens and Lopez-Perez paper proposes one path, using entanglement witness criteria rather than full Bell tests. An entanglement witness is a measurable quantity whose value, under certain assumptions, implies entanglement if a threshold is exceeded. The idea is that even without full Bell test capability, certain observables can serve as signatures of entanglement. The certain assumptions matter. If the assumptions do not hold, the witness loses its power. Warren's critique argues precisely that the assumptions are not justified by the evidence presented. The classical model may be incomplete. The signal may have conventional explanations. The witness threshold may be incorrectly computed. This is the state of the MRI entanglement line, contested with explicit published critique and reply, not settled evidence, not refuted either. A dispute at the boundary of what current methods can resolve. I return to this point often. Not for comfort, for clarity. The uncertainty is methodological, not mystical. We are not looking at evidence that is ambiguous because the universe is mysterious. We are looking at evidence that is ambiguous because the measurements are hard and the interpretations are multiple. Before we proceed, it is worth distinguishing between neural synchronization, a well-established phenomenon, and quantum entanglement, a much stronger claim. Modern neuroscience has documented extensive neural synchronization. Neurons in different brain regions can fire in coordinated patterns. Oscillations at various frequencies, theta, alpha, gamma, can synchronize across distributed networks. This synchronization is measurable with EEG, MEG, and fMRI. It correlates with cognitive states, attention, and consciousness. Hyperscanning studies, experiments where multiple people's brain activity is measured simultaneously, have shown that neural activity can synchronize between individuals during social interaction. When two people converse, make eye contact, or coordinate actions, their brain rhythms can become correlated. A 2022 scientific report study, for example, examined interbrain EEG synchronization during coordinated tapping tasks. These correlations are real and interesting. They are not entanglement. Correlation and entanglement are different concepts. Two systems can be correlated because they share a common cause, receive similar inputs, or interact through classical communication. Entanglement is stronger. It involves quantum correlations that cannot be explained by any local classical theory, even one with hidden variables. The distinction matters because demonstrating correlation is relatively easy. Demonstrating entanglement is hard. Correlation can arise from shared environmental inputs, common rhythmic drivers, social cues, and feedback loops. Two people's brains synchronizing during a conversation is impressive but does not require quantum physics. They are receiving common inputs, each other's words, facial expressions, body language, and their neurons respond to these inputs in coordinated ways. Entanglement would require something more. Quantum correlations that persist despite classical explanation, that violate Bell-type bounds, that cannot be produced by any local hidden variable theory. The MRI ZQC paper claims to have found an entanglement witness. The dispute concerns whether the witness is valid, whether the bounds are correctly computed, and whether all classical alternatives have been ruled out. This brings us back to the core question. What would constitute convincing evidence for quantum effects in consciousness? Now, let us address the strongest counter-argument to any quantum consciousness hypothesis: decoherence. Max Tegmark, a physicist at MIT known for his work on cosmology and the foundations of quantum mechanics, published a quantitative decoherence critique in 1999 to 2000 that remains one of the highest leverage objections to Orch OR. The argument is straightforward. and it is rooted in well-established physics. Quantum coherence requires maintaining phase relationships between superposed states. When a quantum system interacts with its environment, information about the superposition leaks out and the delicate phase relationships are destroyed. This is decoherence. The brain is warm, approximately 37 degrees Celsius, 98.6 degrees Fahrenheit. This temperature corresponds to thermal energy of about 25 millielectron volts per degree of freedom. Thermal fluctuations constantly jostle molecules, disrupting any coherent quantum states. The brain is wet. Neurons are surrounded by and filled with water and dissolved ions. Water molecules collide with any quantum system at rates of trillions per second. Each collision potentially destroys coherence. The brain is noisy. Biochemical reactions constantly occur, releasing and absorbing energy, changing local electromagnetic environments. This is not the carefully isolated cryogenic environment where quantum computers operate. It is the opposite. Max Tegmark estimated the decoherence timescales for brain-relevant degrees of freedom. His analysis considered different scenarios: superpositions of ionic states near ion channels, superpositions of neural firing, and superpositions involving larger-scale neural activity. His result? Approximately 10 to the negative 13 to 10 to the negative 20 seconds. That spans between 1/10 trillionth of a second and 100 quintillionth of a second. To be precise about what these numbers mean, 10 to the negative 13 seconds is a tenth of a picosecond. Light travels approximately 30 micrometers, about 1/3 the width of a human hair, in that time. 10 to the negative 20 seconds is 10 attoseconds. Light travels approximately 3 nanometers, less than the width of a tubulin dimer, in that time. Neural firing and cognitive timescales span approximately 10 to the negative 3 to 10 to the negative 1 seconds, milliseconds to hundreds of milliseconds. Action potentials last about 1 millisecond. Synaptic transmission takes a few milliseconds. The integration of information for conscious perception takes tens to hundreds of milliseconds. The gap between decoherence and cognition spans 10 to 15 orders of magnitude. This is not a small discrepancy. It is vast. It is like comparing the time it takes light to cross an atom to the time since the extinction of the dinosaurs. Under Tegmark's model assumptions, relevant brain processes should be treated as classical. Quantum coherence decays so fast that it cannot participate in neural processing on cognitive timescales. By the time you have thought a thought, any quantum coherence that existed has decohered trillions of times over. This critique does not say quantum effects do not exist in biology.